Algae 11. We're moving on in section 20.4. Plant-like protists that are more of the multicellular variety. It's funny to say multicellular because protists, by and large, you think of as being single cells, but as they colonize and as they begin to form something which resembles multicellular algae, what we've done now is we've lumped in our major algae, our reds, browns, and green into this group. So we'll be considering those algae in the sense of being multicellular protists. I like the term colonial, but it's an interesting group, so let's get started. So, just a sec here. Let me turn on reflection, and we are golden. There we go. Boom. Now, red algae are kind of interesting. If you've ever gone for sushi, when you roll up your sushi, which is essentially a rice, you roll it with a little bit of seaweed. So if you've ever had sushi, you've eaten red algae, particularly a variety called nori. Green algae, well, we see them hanging out in our fish tanks and making a good old mess. And what you're looking at here right now is brown algae. So here's our major groups: red, brown, and green. So let's give them their proper phylum names or divisions, depends if you want to use the plant-like term. Red algae, which is phylum Rotophyta. Then we have our brown algae, which are phylum Phaeophyta. And our green algae, aptly named after chlorophyll, Chlorophyta. And it's this group which gave rise to our land plants. So let's hit them with a nice green highlight. There we go. And let's color code these just for fun. Brown algae. Oops. There we go. And our red algae. There, so we're aware of what we're dealing with. They're named after their accessory pigments, which I had mentioned in 20.3. These are little helper pigments that sort of move photosynthesis along, but they have colors and they change the look of the algae. So let's take a peek. Distinguishing characteristics of red algae. Well, first thing I would say about them is these are the kind of algae that they find at great depths. And in particular, let's make a note, they're very good at harvesting blue light, which is mentioned down here. So let's hit that with a nice blue highlight. Haven't done that yet. Color code it. So they're very good at absorbing blue light. And remember, there's an intense competition for all the wavelengths of light. So if an organism evolved a certain pigment which gave it an advantage, in this case phycobilins, now interestingly enough, that's good at absorbing blue light. Phycobilins are sort of red in nature. There we go. So we should meet our red algae for a second, and just to prove that they're red, their pigment, don't get too caught up on the color. The pigment is red in color, but it's absorbing blue light very efficiently. Make certain, before I show you our red algae, that you're aware, let's go with a nice color like this, turn that down a little bit, that chlorophyll A is the main pigment. It does most of the photosynthesis. It's the accessory pigments, which we've been looking at, specifically our reddish sort of color, which is our phycobilin here, which is making photosynthesis happen at very great depths in the ocean. So now I want to jump out. Red algae, blue light. Let's see what that pops up. Sort of throwing one out to Google here. And let's throw it to images. Okay, so here's our red algae. And these red algae, we've looked at this before, look like this. They kind of look coralline in nature, with this sort of the neat little sort of netting of the leaves and the branching of the plant. This is a bit of red tide, but this here, what we're looking at, our little Nemo friend here, red light. And let's just, red algae, let's try deep water. Because I'll get a nice little diagram of this. There we go. Ha! So they're down quite deep. In fact, further than our green algae would go, which would tend to be near the surface, especially green algae like fresh water. But our red algae can go quite far down. They can absorb this blue light. So they have the photosynthetic pigment, which enables them to absorb whatever light makes it to that depth. Just taking a peek around here. There we go. Interesting little shots of all the algae. Not quite seeing what I'm looking for. Certainly is pretty when you look at algae, though. I'll pop back to this one here. And I'll go back to our photosynthetic pigments, which we had before. Essentially, if you're going to absorb blue light, then you must have a pigment which is an advantage. And the specific one, let's go here. I don't want text. Let's grab a red color. Okay, so if you're going to absorb blue light, the photosynthetic pigment, much like chlorophyll B, I guess in this instant, this is where phycobilin does an amazing job, right around this range here, around the 450 nanometer wavelength of light. Now, blue light, if the way I describe it to students, simplification, but it works. When you go out into the deeper regions of the ocean, think about what color it is that you're seeing. You're seeing a lot of blue. It's very blue water. Well, the only wavelength that can really focus and make it into the depths of water down to where red algae are is around blue, about 450 nanometers in wavelength. That turns out to be an advantage because red algae can live where other algae cannot. So they've essentially got what you would call a niche environment. Let's pop back to 24. There we go. So they have a deep water existence. Okay, brown algae. Now, brown algae, if you live off the coast of British Columbia, you go down the west coast, go down Oregon, south, off the east coast, brown algae is very ubiquitous. First thing you should see is what brown algae looks like. Brown algae is often known as kelp. If you've ever heard of kelp, rockweed, you've seen brown algae. At low tide, you'll see it right up near shore. Oh, heck, even in high tide. This stuff, there we go, is sort of slimy, and you'll find it on the beach. Sometimes you'll find these kelp whips. There you go. There's fucus or rockweed right there, and you'll see it right near shore. This is the variety that I played with quite a bit as a kid, right? And just, it's incredibly successful, but it's the kind of algae, it has a long stem and it attaches to the ocean bottom, kind of glues itself onto the bottom. It can't be that far out. Not the variety that we see around here. So the brown algae are very successful, but they're more of a sort of a shallow water environment. That's what they like. So our brown algae have an accessory pigment known as fucoxanthin, and we highlighted that in brown before, so we'll go back to brown. There we go. Fucoxanthin. They have chlorophyll A and C, which are green and sort of limey yellow sort of colors. So let's pick a nice sort of green color to delineate those. I'll choose this. But not quite that strong. Need to save these as presets. There we go. So they've got their chlorophyll A and C, but then the accessory pigment really makes the magic happen. It's a little helper for the action of chlorophyll. And remember, what chlorophyll is doing is it's capturing energy from the sun and helping carbon dioxide and water to be brought together as a sugar that we know as glucose. So brown algae, they're very big. In fact, you get these kelp forests, and there's a famous one, famous kelp forest you might know or may have heard of, called the Sargasso Sea. There we go. And that's off the east coast, a tremendous source of brown algae. So we can find them. Here we go. The Sargasso Sea, zoom in here. So you've got the Atlantic United States, and as you go out into the mid-Atlantic, hurricanes come through this region all the time, and you can see Bermuda's in the center of this. There's this tremendous brown algal growth all in this region here, and you can see it stays extremely well mixed, and the nutrients are constantly being delivered to the brown algae just through the natural ocean mixing. And you get this thing called the Sargasso Sea. It's quite fabulous. Here's another shot of where you would find it, just represented by that black oval. The Sargasso Sea is just tremendous floating mats of brown algae. Not every variety of this algae has to necessarily be attached to the bottom. That's what we tend to see off the coast of BC. But out in the Sargasso Sea, this makes a very interesting ecosystem for not just plants, but overall biodiversity for animals, etc. Because there's a good shot of it. Look at that one there. It's a great place to hide if you need to get away from predators. Let's put it that way, right? Nice and murky. I like to think, when I think of brown algae, by the way, of not jelp, but kelp. If you're from BC, this is probably the variety that you'd be most familiar with. And you'll see these kelp whips. There you go. Now this is brown algae, and what's left, what really you're looking at, that long whip-like structure, is basically the stem, and the leaves float on the surface of the water, and at the end of the stem, what you can't see is a root-like structure called a holdfast, which is let go. Now, I was awful as a kid and let my brother have it with one of these, older brother, and he'll never let me live it down, but these do function like whips. Don't pick them up and use them like this. It still functions like a whip, where the idea is you'd send that tremendous velocity down, right? Down to the tip. Kelp whips can be good fun, but it's just it. You don't want to mess with this too much. And yes, it did cause quite an injury, but that was just revenge on an older brother. Not that I condone that or anything. Anyhow, well, I'll confess, I was in grade 7. The brown algae are multicellular, so we're getting to a point here where we've lumped these brown algae in with, they're not quite like our land plants. In fact, those algae, when you look at the leaves of them when they're floating on the surface of the water, we'll see this in a minute, are only about one cell layer thick. So for convenience more than anything else, they've lumped the multicellular protists and into the protist category and called them algae. There's got to be something between colonial protists and plant-like things. So the algae are kind of being lumped into the protist category here. They're calling them the multicellular ones. Again, what we're looking at is let's go here. Let's go with a nice sort of light green. Turn that down. Haha. So, oops, wrong one. Back to blue. There we go. What we're looking at are multicellular. They're calling them protists. Colonial protists had to give rise to something multicellular, so we're lumping the algae in here for convenience. But if you think about the algae in this group, they're just basically multicellular protists that are hanging out in cool, shallow waters off temperate or Arctic areas, just as they say in this slide. Now, this one will always be on a test, so let's call it for what it is. A test item. Blades are basically the leaves, and by blades we're talking about these items here. And they're only about one cell layer thick. And their job is to do exactly what leaves would do, to photosynthesize. In the ocean, you don't really find cell walls. These things don't have really cell walls. They're flimsy, and they float on the surface of the water. Pretty basic. Your bladder Your bladder's basically a float device. It's filled with a gas, and it's basically what holds the plant up at the surface of the water. The stipe, we could just as easily call the stem. And the holdfast, it's root-like. It's not really a root because it doesn't do any absorbing. So we'll call it a root-like structure. Roots haven't really evolved if we're thinking about this story of Biology 11 as we're going through it. This more or less glues on with like a calcium carbonate like glue. And what causes the algae to be torn off from the bottom, you think that's pretty strong. Well, there's little sea urchins and things that will chew the holdfast, and they'll eat it, and then that causes the kelp to just come loose and basically float away. Major storms, they can tear this right off the bottom. This is continually happening because of what you find up on the shoreline, especially if you go off the coast. You'll find lots of brown algae. Interestingly enough, they show this here. Fucus, when you look at it, just so we're clear, fucus is known as rockweed. And that's not what they're showing there. They're just showing your average kelp. But fucus known as rockweed looks like... Let's get a good shot of it. Yeah, about like that. You can see all the little gas bladders, and you can see the blades sitting up high. This is what fucus really looks like. Sort of this variety. And this, it's a little bit of an oversimplification what they've shown you. There's a field of fucus there. So what you're looking at here is just sort of a large, long sort of section of brown seaweed, if you will. This isn't the genus Fucus, but it's made up of, you've got your holdfast, and you've got your stipes, and your bladders, and your blades, and everything, like I've said, the holdfast holds the algae to the rocks. So there we go. Remember that the main photosynthetic pigment with these is fucoxanthin. Now some definitions here, which we've already covered. You've got a stipe, which is like a stem, leaves or blades, and the gas-filled bladders, they're necessary for essentially flotation. You have to keep those leaves up on the top so that you can photosynthesize. Neat thing about this video, if at any time I go too fast, you can pause, rewind. I'll leave the controls up to you. Try to be aware of your pace. Green algae are essentially what gave rise to the land plants. Our little friends here are known as the chlorophyta. We've made that distinction before, but I've got lots of space, so I'll put it in here. Okay. I like to think of it as the green phylum. Okay. So what are the distinguishing characteristics of our friends? Okay, so these are the most like our common land plants. In fact, our green algae are very much freshwater, and they gave rise to things that hang around freshwater. Things like the mosses. And if you've ever jumped off the end of a dock and you look at these little sort of fuzzy-tailed things under the water, okay, you'll see things that are called hornworts. And some fish tanks have plastic versions of that to keep the fish feeling at home. So we've got our green algae, our mosses, our hornworts. Ultimately, what will happen is that land becomes an advantage, an opportunity, if you will, for plants, and they really couldn't say no. The ocean is the mothership, but there was opportunity in them there hills, so plants went in and took advantage of it. So if you think about plants that you know that are always associated with water, mosses and hornworts and things like that, you pretty well see how this sequence played out. When we look at green algae, the cell wall composition is very much cellulose. It is as close to what we'd expect to see as we find in land plants. So it is a hypothesis because nobody was there, but they're pretty darn certain when you layer out the characteristics. You look at the photosynthetic pigments, chlorophyll A in particular, chlorophyll C, cellulose cell wall. As these characteristics are matched up with green algae, you see a lot of overlap. And that's led to the hypothesis that the chlorophyta would represent the closest ancestor that's still in water to our most primitive land plants. It's all sort of part of a grand analysis, but it is just a hypothesis. So green algae you can find you can find in fresh water. You'll find them in salt water too. Um, just try to keep green algae out of your fish tank. I'm almost scared to introduce those algae wafers, especially with my little algae eater, because eventually what will happen is I'll have green algal contamination. It gets into the water filter, and they just love it because the animals in the tank are releasing all sorts of fertilizer in the form of feces and nitrates and things like that, and that's good for plants, and the plants will just darn well take advantage of it. Now, interestingly enough, most of the green algae, when we sort of we look at this and we say green algae, is it a protist? Is it unicellular? Is it multicellular? When you look at the green algae, you find some that live their lives as single cells, but they can colonize. And then we see other green algae, like you'll find in the fish tank, which are really colonial. At which point we say, boy, do they ever look like they're multicellular? They're always together. So we say, you know what? They're pretty well multicellular. Pretty well multicellular is the reason that they were taken out of kingdom Plantae, and because they're very much resembling protists, we put them in the protist category and just said they're so colonial. They're like single protists that are colonizing so much that we're just going to put them over there because they are still quite different from plants. So that rule that protists can only be unicellular isn't quite enough glue to just create exclusions because the algae should be fit in this group, right? Especially when colonization is a common feature in protists. So how do these multicellular algal protists reproduce? Well, they have a life cycle where they'll be diploid, and diploid is sort of like you and I. Diploid means that you are 2N. Means that you've got a set of chromosomes from dad and a set of chromosomes from mom. And then there's what we call a haploid generation, which just has one set of chromosomes, one set of DNA. So when you look at algae, they do an interesting thing with their life cycle where they're going between a period where they are diploid with two sets of chromosomes and haploid where they only have one set of chromosomes. So we'll get into that now. Now, a topic comes up now that's called alternations of generations. It only applies to organisms that are multicellular. So let's make an important distinction here. Multicellular, and there are two versions of the organism that are necessary to complete the life cycle. And what we'll be seeing is a shift between a sexual version and just a version that clones itself, an asexual version. So it's important to make a little distinction here. Shift between those two modes. Now, I think what I'll do, we'll talk about Clammy de Monas first, because Clammy de Monas, it's right on the verge of alternation of generation, but Clammy de Monas lives its life as a single cell, so it doesn't quite qualify. Okay, so Clammy de Monas, if you've ever seen SpongeBob, by the way, you might know this character. known as plankton. What a little evil genius. As I talk about Clammy de Monas, I want you to think about our friend plankton, because as far as I can tell, plankton is an example of Clammy de Monas. Our little evil genius here has a red eye and he has these little feelers. I'll explain what these are as we go along. He's a single cell, just so you know. And entertainment, my daughters like this. They often portray him as a multicellular organism with teeth and things like that, but sorry to burst the bubble, that wouldn't be possible if he was a single cell. Wouldn't have all those special tissues. So getting back to Clammy de Monas, it spends most of its life in the 1N stage. Curiously, haploid. So let's do a note here. It likes to be 1N. Now remember, the only time you get 2N is as a result of sex, bringing together genetic material. So I want you to keep that in mind as we move through, basically through our slideshow, because you'll see that the purpose of sex is to shuffle the genetic deck, more or less to speak, to introduce some variation, to come up with something new for the next generation. So reproduction in Clammy de Monas sort of forms these two life cycles. Well, not two life cycles, two specific cycles. One delineated by the orange that you see right here, there you go, is the N, or that's understood to be 1N, one set of chromosome, part of its life cycle. And you see from the orange color, if you look closely at these circles, it is in the haploid phase almost the entire time. The only time it's going to become diploid, right here, let's switch colors, because that's a purple color. The only time it's going to become diploid is when it decides to go through a sexual phase and form a zygote. And sex happens in the animal kingdom. The need to introduce genetic variation is when things get tough in the environment. They're tough times. And these cells, maybe the food source isn't as available, or the cells are stressed, and they decide to have sex, produce variations of themselves so that those might have a better shot at the next generation. So most of the time when we look at Clammy de Monas, and we'll just zoom in on this side, the cell, and you can see why it looks like plankton, right? There's the little red eye spot and the two feelers, and right? Our little friend plankton here would just prefer to go through mitosis, which is like cloning. If you ever cut your skin and the cells grow back, they just clone in, right? There's nothing new. It just replaces what's already there. And plants do this all the time. If you've got a spider plant, you'll see a little version of that spider plant growing off the side. And kind of interesting, why is it growing another little version of itself off to the side? Well, it's a way of reproducing, creating other versions of yourself, but they've just got the same genetic makeup. So if the organism's doing good in the environment, it will just go through this cloning phase of itself and just reproduce itself through, in this case, what we call zoospores. And there we go. So this right side of the life cycle will just keep going and going and going. However, just like anything, can't expect things to stay the same. If the environment isn't so good, the organism would be triggered to go to another life cycle in which it would produce some specialized, what we call gametes. Now, they're showing them as plus and minus. You can think of that as male and female. They're just opposite. So we get the production through mitosis of some little gamete cells, which will fuse, and that's what you're seeing. Let's switch our color now. Actually, I have to hit done. Our gametes are fusing here. So that essentially is, you can call it symgamy, you can call it fertilization. The genetics are coming together. Each one of these has one N, one set of chromosomes, one set of chromosomes, and at this point, as they begin to fuse, you get a 2N zygote. And it's right at this point when they fuse. So this guy here is 2N. Now that's just a shuffling of the deck, and what you'll notice is that Clammy de Monas does not stay there. In fact, it goes through something called meiosis, and that's the same process that's going on in the testes of, we'll just use human males as an example, to produce sperm. The testes themselves are diploid. That tissue has two sets of chromosomes, but what it does in a genetic shuffling of the deck is it's picking one half of the genetic material and just coming up with combinations of genes. But meiosis always produces a 1N cell. When you look at the zygote, this sort of thick-walled zygote will hang out and wait until conditions are more or less ripe, and then inside of it, it will undergo meiosis and it will release new versions of our friend plankton, haploid cells. And that is what you are seeing here. And those are free to carry on the life cycle. They can either go back into the cloning phase, or, depending if times still aren't that great, we may get the production of gametes again, where the two versions of Clammy de Monas, Mr. and Mrs. we'll just call it Mr. and Mrs. Plankton, come together. They make a baby, in this case a zygote, and that zygote waits and regenerates the haploid cells. It's an interesting life cycle. When you look at it and you zoom out on it, what you should see is that the cloning side, the haplontic version of the life cycle, is essentially, let's draw a line, is essentially here. So this haploid life can be described as haplontic, and this specific moment where it's producing a zygote, the whole purpose of that leads to a diplontic purposed phase of its existence. Shuffling of the deck. If you don't need to shuffle the deck, if you're winning every hand, stay in the haplontic side. But if you do need some variation, some changes, go to the diplontic side. As I say, this isn't alternation of generation formally. It's close. It's close. The ensuing slides just are more or less a summary of this. So I will display these for a second, but they're just the same information over and over again. This is the cloning side with the zoospores, self-regenerating. Mostly just mitosis on this side. Notice you don't see meiosis. Meiosis, which meiosis takes a 2N down to a 1N. Takes a diploid tissue or organism to its haploid equivalent. This is completely asexual. No variation. It has nothing to do with variation. Just push this along. Conditions become unfavorable. Clammy de Monas goes into sexual phase. And we see this side with the production of the zygote. What you would need to take away from this is the reason why this is happening. And you would need to take away that there's a haploid and a diploid existence going on here. It's important to know the why so much more than the small details and everything in these diagrams. That's much more testable that you understand what's going on, the reason for what's going on. There we go. Interestingly enough, when our little friend Plankton here decided to produce cells that were going to come together and fertilize, because the cell was already haploid, it's already 1N. sort of a side note here I want you to realize. Our friend Plankton over here was 1N. Well, he had to use mitosis. Once you're 1N, you can't really go any further down than 1N. So when he produced sex cells here, it was by mitosis. The only time meiosis will occur is with something that is diploid, 2N. So make a note that these cells that come together to have sex had to be produced by mitosis. It's all that's available once you're 1N. Then here, when you've got your 2N zygote, 2N goes to 1N, and that's the only time. That is the only time you can do meiosis. For everything else, we have mitosis. So at this point, what you should probably take away is that Clammy de Monas really likes being haploid. In fact, that's its favorite thing. It will get back to that haploid existence as fast as it can. It only does this for a very brief portion of its life cycle. What's being described here is that thick protective wall is a great place to hide out when times are tough, and there can be some really nasty conditions. Clammy de Monas is basically almost in a state of, I wouldn't call it suspended animation, but I would call it a resting phase. And when times are good, food sources are available, maybe there's nitrates in the water, some iron content, times are just about ripe to pop out, the zygote receives that stimulus that it needs to grow, divide, and it has, in order to develop the haploid cells, remember, and I'm making this quite the point, that it's doing this by meiosis. And we get back to our 1N plankton back in the middle. There we go. A little bit of trivia here. Each zygote produces 4 haploid cells. So each one of our little friends here is 1N. Zoom in and put that in. There we go. I like that zoom feature. It'd be pretty hard to rate that otherwise, even with this neat little stylus that I use. Moving into proper alternation of generation. I had said that you have to be multicellular for that to happen, and that is the formal definition. And I often think of the movie Alien for some reason for this, because it's like you've got 2 different creatures, right? If you ever saw the movie Alien, quite the horror movie, don't let your little kids watch it. But when you get older, I guess it's fair game. You've got that version that jumps on the person's face, and then you've got the version, unfortunately, which comes out of the person. What you're looking at, in order to complete the life cycle, is you've got 2 different versions of the multicellular organism. So you have to look a little closer. There's an alternation going on of generations in order to complete the life cycle of our critter. It turns out that one little multicellular green algal cell, or green algae, does this, and it's called ulva. Ulva has one version that produces gametes and the other version which just clones itself. So it needs one version of itself to go through the sexual phase of its life cycle, and it's rather interesting. Let's sort of just pop ahead to the next slide and you can see this. But there's a gametophyte version of the plant, and what you're going to see coming up called the sporophyte version of the plant. So here it is. Let's zoom in just for a second. We don't need the details. I want to point out with, let's grab a different color. That's a little bit more, green won't work. Let's grab nice dark blue. In the center, you see three things that look like a large green leaf. This one at the top is diploid, and these ones at the bottom are haploid. And you can think of one version as being female and one version as being male. So what we have is an alternation. It's kind of odd because if you look at sea lettuce, you wouldn't know which generation you're looking at. Are you looking at the 2N sporophyte, or are you looking at the 1N gametophyte, the gamete maker? Let's check it out. Sea lettuce, ulva, is the genus. There we go. Looks like this, and you can't tell whether or not you're looking at the 2N sporophyte generation. We call it the sporophyte generation because it produces the spores, or if we're looking at the 1N gametophyte generation, which is the one that produces the male and female gametes. You just can't tell. Now later on, as we study different plants in this course, especially moss is a really nice version because you can tell the sporophyte from the gametophyte. The sporophyte will look different than the gametophyte, but in some of our earlier examples, you just can't tell. It's just a green leaf. So you've got quite a life cycle to look at here. So what I think I'll do is I'll take this stepwise and then we'll come back to this diagram. This is alternation of generation. So let's put that in. Let's see if I can zoom down on this a little bit. There we go. Like so. So if we start with the gametophytes are the 1N version of the plant life cycle. And what we can see is that it's a fairly lengthy part of the cycle. In fact, this being the haploid phase, where it's 1N, is fairly lengthy. And what will happen is around the proper reproductive time, the female gametophyte will release something which it's not quite, you would expect it to be an egg, but at this point, it's just much more simple. It's just a flagellated gamete, and it can cruise around in the water. So the gametes, depends which one you're released from, the male or the female, the gametes will get together, and you can see why ulva, even though it's multicellular, you look at these gametes, and the gametes look protist-like. These little flagellated gametes come together, and when they do, we get fertilization, and at that point, we get something that's diploid. So this is typically what you would expect. And in fact, you could think in the human equivalent, when the sperm and the egg come together, that's fertilization too. But the difference is, humans are diploid. These plants are haploid. And you can see how that's a little bit of evolutionary heritage from our friend the protists. Okay, so what happens? In the diploid phase of the life cycle, let the highlighting begin. So the gametes fuse. And got that. And of course you get a diploid zygote, that's normal. Zygotes are always diploid. What you get is let's switch the colors here. You get the diploid multicelled version of plant, which is ulva. So back to our colors here. This version of the plant may look like the gametophytes, but this one has two full sets of genetic material. And if you ask yourself the question, well, why did it mix the genetic material? because it's looking to introduce a shuffling of the genetic deck, right? Remember back to our chapter on when we were studying Darwin and we were talking about genetics. Mutations are, I like to say, mutations are like new cards in the deck, and sexual reproduction is a way of shuffling the deck. Well, good populations do want to eventually, with through mutations and through the selection process, you want to get these new versions of genes sort of throughout your population. Best way to do it is by sex. So the gametes fuse here. You get the 2N zygote. Now you're at the sporophyte stage. You had to ask a question, well, where did the gametophytes come from? It's a large game of chicken or the egg, right? Well, the 2N sporophyte does exactly that. It produces spores, and what it'll do is those spores are haploid, and those spores will reproduce the gametophyte generation. So you have to know, oh, here it comes. Let's move that other way. Meiosis as a process is about to occur. When the spores are produced, they would have gone through meiosis, and you've got just a 1N thing drifting down towards the bottom, which is here. So ova undergoes meiosis, and you get these little spores, and if you look at them, they're flagellated. Well, why are they flagellated? Because they're going to swim down and find a nice free spot on the ocean bottom where they can grow and reproduce. So each spore, as I say, can grow into a new individual. They don't fuse with anything else. These spores are intended to become the new gametophyte plants. So because the 2N version of the plant made these spores, we say that that diploid phase is known as the sporophyte. It's worthy of a highlight, don't you think? Diploid ova produces spores, therefore it gets called the sporophyte because it's a spore-producing organism. So you're going to see this trick where the sporophyte regenerates the gametophyte. So around and around we go. Sporophyte, gametophyte, and that is what alternation of generation is. We have the sporophyte version and the gametophyte version. Okay, coming up on the finish here. I've sort of spoke at length about algae producing oxygen, and not only by producing oxygen does it take care of the planet. Oxygen, as it drifts up and interacts with UV radiation, becomes ozone, and that protects us from obviously the nastier parts of that kind of radiation. Otherwise, we'd be developing skin cancers, melanomas, and worse, because radiation is a very intense thing to have to experience. We also find algae in sushi, ice cream. Algae is an emulsifier. And in order to emulsify something, if you haven't heard of that before, that's when you take something fat-like and bring it near something which is water-like. Fat and water, they generally don't mix. But if you add an emulsifier, if you've ever done this, a little bit of egg yolk makes a good emulsifier. You can mix oil and water together. So think about ice cream. Lots of fat, lots of water. So algae turns out to be a good bridging sort of material for ice cream. And I wanted to show you that if you like sushi, you've been eating red algae for quite a while, the genus of red algae that you consume with when you have sushi, by the way, the rice is sushi, so anything you make with it is referred to as sushi. These mats you're seeing here, you can sort of see the one behind. You see that, you can see its strong reddish tone, right? These are red algae, and there you go. You can see the reddish color here. So next time you eat this, ask yourself a question. How much sushi have I eaten in my lifetime? That's question number one. And number two is, could stuff like this be used to solve world hunger? There's a lot of edible seaweeds that we're simply not taking advantage of. So this one in particular is my favorite. Thank you, red algae. And the list goes on. What else can you use algae for? Well, it turns out that without them, we'd have a hard time making plastics, transformers. They're in our deodorants, paint, lubricants, artificial wood. And when we were doing some of our work in the lab, we played around with something called agar. And agar is just, I like to say seaweed jello because it's about the most accurate term. Agar in our Petri plates, you guys saw as jello, not agar. I meant agar. Darn fingers. Here we go. So this right here shows how our lab tech, Mrs. Giesbrecht, produced this. So it starts out in a liquid form. It's basically just a powdered material like jello, and then we pour it into the Petri plate, and it solidifies and becomes a nutritive surface for us to do our work with bacteria. Bacteria love it. It gives them a lot of the nutrients they need. We can grow all kinds of crazy things up on it. And they've been, oh, there you go. Maybe if you want to make yourself a nice jello mold like that. But agar itself is just powdered seaweed. And that's it, folks. So that's 20.4. All that really remains is to talk about the probably the most cryptic little critter here, which is the trying to come out with it here, the fungal-like protists in section 20.5. Now, here's a piece of advice. If you want to run through the Clammy de Monas life cycle, you want to check out a little alternation of generation, here's what you should do. Let me just pop out of reflection. On the course website, we put some pretty neat stuff in here. Now, this is the Plantlike Protists. Click on this and you'll see the Clammy de Monas life cycle is waiting for you right there. Click on that, you get the larger version, and you just click on Start Animation, and it'll talk you through this, and there's even a little quiz at the end, and it goes into more specifics than I did. That's a good one to do a little guess and check and sort of test yourself out there. Oop, lost it. The second item that I had there, I've got Clammy de Monas shown twice, just a little bit more information. Here's a neat one, little flash video on ulva or sea lettuce. Now I'll just drag through and go through this pretty quick. There is a narration. It gets into quite a bit of detail, but what it does show as it pops through is that we get an alternation of generation. Here we go, which is the Gametophyte, and these are often referred to as the mating type. Nice little summary on the side, and that they will produce our specialized structures, our gametes, which will come together and regenerate our diploid sporophyte right there. So great little animation, comes from Kids Web, and I'll let you check through that. But as I've said, there's a great number of things that you need to go through on here. Some great YouTube videos that I've shown you, and there are more here as well. Okay, so I'm going to check out. That's 20.4, where we talk about the multicellular protists, which I think is an interesting term, but I think now you understand that the protists cannot just live in a unicellular existence, but they do live in something which is exactly on the borderline of a multicellular existence. Okay, but remember, protists can live at various parts of the life cycle in a unicellular way. Nothing else can do that, and that makes them unique. And protists, because they can be plant-like, and they can be fungus-like, and they can be animal-like, are very good at looking like the other ones, but because they're not multicellular, pretty well all of the time, they don't live a multicellular existence. That's why they don't fit in those kingdoms. All right, ladies and gents, have a good one, and we'll see you later.